Hello, everyone, and welcome to another super exciting educational webinar.
Recording in progress.
Today, we are here to talk about fungal fun, to have some fungal fun with Peter McCoy, Dr. Mary Cole, and Dr. Elaine Ingham. And I am Dr. Adrienne Godschalx. I am here and excited to have a fun-filled fungal conversation. It's a great tongue twister for you. So today, brief overview. I have a few housekeeping things to share with you about how to engage with the Zoom webinar if this is one of your first. And then we get into having a panel discussion. I know these folks are excited to engage with one another and learn from one another. And then you get an opportunity as well to share your questions. So your questions, we have a couple options in this webinar platform. The Zoom live chat is a place for you to come and share where you're joining us from today. Please welcome yourself. Say where you're coming from, what you're excited to learn, and welcome to this conversation. So excited to have you here. So yes, this live chat is a place for panelists to interact with one another, for us to get to see you. We have folks coming in from Canada, Florida, New York, Sweden, Belgium, Brazil. We caught a lot of time zones. This is exciting. Actually, it was carefully coordinated in order to get Dr. Mary Cole in Australia in a time zone that works for us over here in the Pacific Northwest. But we also caught some UK, Central California, Colorado. Oh, Vashon, nice, close.
Great.
I think I see a few of my students' names from all over. Oh, the Yucatan. Awesome. Where the meteorite hit Earth. Awesome. It's a great location.
Scotland. Texas.
I always love this part, getting to see just how global our thinking is about soil and these organisms that are collaborating with us to keep us alive. Port Townsend, Gainesville. This is exciting. So yeah, stay connected to the chat and welcome.
Did I see Santa Claus from the North Pole just go by?
That makes sense. Yes. Happy holidays, everyone. Ho, ho, ho. Clever. I like it. And we also have another feature, the Q&A button, which is right next to that chat, which if you have a question for our panelists, that is where we will get to see the questions coming in and be able to address them in conversation. So start piling in your questions. Oh, look, here's a graphic. How convenient. So yeah, live chat. Many of you have found it. And the Q&A button looks like this both-way conversation so that we can engage with you. And before we get rolling, we have a quick poll, partly so that we can understand who is here, who is curious. Are you a farmer, grower, or ag professional? Are you an environmental advocate? Do you fall under both of those identities or neither? All of them are great. Wait for the poll. I see you're entering in the chat. We will have a poll pop up here in a moment, I believe. Ta-da! And you can make your choice.
It helps us understand exactly who we're going to be talking today, so we might change things a little bit from farmer, grower, ag professional, or more environmental advocate if we're strongly skewed one way or another. So how long are we going to let that go on?
It's a great question. As folks finish up, I am going to start to introduce our panelists today. And as always, it is my great honor to introduce Dr. Lane Ingham, a pioneer in understanding that fungi aren't just there in the soil, as her committee told her back in graduate school in the 1980s, but in fact are a fundamental component of soil. And so, welcome, Dr. Lane. We also get to have Peter McCoy joining us today, who not only wrote Radical Mycology, which I realized today I have featured. So Radical Mycology, which is both a philosophy and a text and an introduction to working with and collaborating with fungi, as well as a school, MYCOLOGOS, which you'll hear more about. And so welcome, Peter. We also have Dr. Mary Cole coming to us today from Australia, who is a delight and just has a way of helping someone embody the experience of what it feels like to be fungi when she talks about them. So I hope you look forward to that as well, because I sure do. But she is over in Australia studying plant pathology and how we can work with fungi and manage plant pathogens in a way that can help us grow plants healthy. And my name is Dr. Adrienne Godschalx. I am coming to you from the philosophy standpoint of being a chemical ecologist, that it is the metabolism of each organism that empowers them to be exactly who they are and to form their relationships in this world, and that chemical and phytochemical and mycochemical diversity is what makes this world so beautiful. So let's jump on in. All right, Peter McCoy, I will give you a chance to introduce yourself and tell us a little bit more about yourself.
Well, thanks for having me. Yeah, I live in Portland, Oregon, born and raised in this part of the world, sort of steeped in fungi growing up, but didn't discover them until I was a teenager by happenstance. And luckily so, got the itch, got the bug, got the spore, I don't know. And that kickstarted was sort of the first domino of the last 20 years of being interested in these organisms, initially from a cultivation standpoint, just trying to learn them to grow them. As a teenager, alongside the vegetable garden, I was growing in my backyard, family's backyard, and eventually pretty quickly got into being curious about their biology and their ecology a little bit at the time, as best as I could understand it. And then eventually, as I got older and learned more and got more concerned about environmental and affairs of the world, started thinking more about how mycology as a science could weave into those topics, and recognized sort of indirectly that nobody else was making that connection and all the things I was reading and learning about the world, and that mycology was always absent. And so as I got a bit older, I started to formulate thoughts around how, and tried to figure out dialogues or conversation points, bullet points, talking points around how mycology should be woven into topics on environmental protection. Why not, right? Even though nobody else was really saying that. And so that evolved over a lot of my 20s in expanding those conversations in a myriad of ways with many different people, trying to just think about how to not only contextualize mycology, make it interesting, really was the hard part, or more engaging or more accessible, I guess is a better way to say it, but also really relevant for people living in our modern life, especially coming from a background like I did, where I was never told that these organisms matter whatsoever. I wasn't even told about them. So why would they even matter if I wasn't even told about them? So a lot of my work has really revolved around, on one end, learning about mycology on all fronts because it's so fascinating from every angle, and then trying to translate that and make that accessible, but also interesting or again, contextualized for the given audience, so that we put these important organisms more central to conversation, in whatever paradigm, whether it's from a creative standpoint, from an environmental standpoint, from a food production standpoint. They certainly have a place in the conversation, even if you might not have heard that your whole life or in all of your education, probably just because your teachers didn't even know how to talk about them. So it's really sort of catching up what we've missed out on. Looking, a lot of my work is try to look at how to close those gaps, increase mycoliteracy, as I like to say, culturally, and create more awareness and appreciation. And so my work has meandered in a mycelial way over the years, just sort of following what is most exciting to me, sort of sometimes in the moment or in that period of time, from say the micromediation work, and really wrap my head around that and really understanding its constraints and limits, rather than it being this thing that can solve everything of pollution, and really wrapping my head around how to make that accessible. And say from a grassroots approach, where I was originally really heavily promoting it many years ago, to over the years getting personally more interested in, say, the cultural history and implications throughout the human story of fungi, and bringing that up to people's awareness, just to bring that connection to us, and then along the way absorbing everything I could about fungal ecology and all their applications as well. So I now currently, as mentioned, not only have translated a lot of that information into my first book, but now have been working for the last several years of translating those tips of icebergs that are in the book into much more in-depth courses that go into these and many other topics. So that's done through my school, MYCOLOGOS, which is in part a mushroom farm. Actually, behind this wall is my fruiting room. So I also run a whole organic certified mushroom farm. Right now, we're primarily online based, as most things are, but the hope is soon enough to be doing all kinds of in-person and immersive trainings on these topics and many more. So I guess I'll leave it there, but that's a bit of what I get into.
Wonderful. Thank you, Peter. And Dr. Mary Cole, welcome. Thank you for being here today. Can you please tell us a little bit more about yourself?
Well, I guess my introduction to fungi started when I was about 4 years old. My father was not a trained scientist, but he was a naturalist. And he took me out into the forest looking for mushrooms because we foraged a lot in my younger life. And there was a 4-year-old me being introduced to a mushroom, being told that, just look at this one. We don't collect that one because don't you see there's a family living in it? And then we'd go on and he'd say, now we can collect these because these ones don't have people living in them. And I guess it wasn't until I became a teenager and older that I realized that my father was actually giving my 4-year-old self my first introduction into which mushrooms were edible and which ones weren't. And I thought that was a really nice way of doing it. So I still look at a mushroom and I say, oh gosh, there's a family living in there. You know, I definitely can't touch that one. And so then as I got older, I realized that as we were foraging, we were finding fewer mushrooms in the fields. And it wasn't until I came to Melbourne, I went to Monash University, I was working with my mycology professor, that I realized that there was an increase in chemicals being used in farming. And parallel with that, there was a decrease in the fungal flora. And so there had to be a connection. So I went through my PhD and then at the same time had this interest that if the mushrooms were disappearing from above the ground, then something was happening below the ground. And my classical training in mycology was in plant pathology and in both the micro and the macro fungi. So there was a parallel interest there in what was happening below the ground and how that affected plant health. But at the same time, I was also a consultant to the Poisons Bureau. So I was getting a lot of information and having to keep up with my macro fungi through the inquiries that were being made through the Poisons Bureau and identification of mushrooms. So my father and my 4-year-old self came in very handy in that particular aspect. I then, through my academic career, it was all in fungal taxonomy, plant pathology, and therefore that continued interest in both again, the above and the below ground. What was interesting, though, more recently is this, well, the potential that, well, it's happening in Australia. I'm not sure if it's happening around the world, but there's now soil health is becoming mainstream here in Australia. And also the impact of climate change is having farmers look at another way or an extra way of being able to remain on their properties and keep the properties resilient. And that's with carbon capture. So there's a lot of work now being done on capturing carbon into the soil using organic matter. But as you all know, probably, that the fungal component is one of the major in-soil biomasses that helps to capture and hold carbon. So I'm doing a lot more work now with how to improve the soil quality for farmers so that they make their farms more resilient, so that they're able to withstand the impacts of changing climate, but also to maybe get another income stream through carbon capture. And what was really interesting, just yesterday, and I'm still at a level of excitement that may be obvious or not. My little company, Agpath, and myself and a colleague from Melbourne University, we've just been given a $300,000 grant to investigate mineralization and quantify mineralization and biological status in organic amendments in soil. And so we're incredibly excited about that. And it's all new news. And so what we're going to be able to do is what I want to try and quantify is the role that particularly the fungal component of the soil and compost, and quantify that and actually have figures around that or data around that will help people more understand that there is an important microbiological component to adding composted and any sort of raw material that can be composted into the soil to improve soil quality and the whole soil biota. And I mean, Elaine and I have been doing this for 40 years, but it's trying to have some data that people who are still not quite sure about it, giving them that information. And so I'm open now to people around the world interested, and particularly in Australia, who would like to contact me with this project over the next 2 or 3 years, and to actually improve the understanding that the fungal component of the soil has in improving all plant health, all soil health, human biome, the whole thing. The fungi are so important, and I want to make them front and center.
As they are. I have several students in Australia who I'm sure are going to be very excited about that proposal and will be in your inbox before you know it.
Good. And before I finish, I really would like to acknowledge that I'm speaking from the Bunurong and the Wurundjeri traditional owners' land. I would like to extend my welcome to elders, past, present, and future, and any First Nation people around the world, actually, who are listening to this. Welcome. And I have a privilege of living on this land, the Bunurong and the Wurundjeri. And the top two photographs, well, the top photograph on the left is my farm. The top photograph on the right is my neighbor's farm. And I would really like to talk about that because there is fungi in action.
Brilliant. Go ahead. Let's hear it. You can do it now.
Ah, well, okay. Well, just the photograph. This may be a little bit out of context, but I've been on my farm for 48 years, my husband and I and our little dogs, and we have never used a synthetic chemical on this farm. We have 60 acres, about 24 hectares. We have always used fish and kelp and rock dust, molasses, any sort of composted material. And we have run a stocking rate where we're able to move them around on a regular basis. And we make compost tea after a visit from Elaine back about, 2011, Elaine, thereabouts. And Elaine and Alan went out and made a compost ring in our garden. And ever since that, Alan has when he wants a little bit of therapy, he makes a compost ring. And he makes the most beautiful compost. It goes all around Australia. He makes compost tea. Now, the farm that you can see now, that has compost tea put out probably 2 or 3 times a year. Now, one of the things you'll notice in the background across the fence, there is yellow. The picture on the right, full of capeweed. Now, capeweed is a flatweed. It's a nuisance. Farmers get out in the spring and they get their cans of glyphosate, and the departments of agriculture tell them to put out their glyphosate and spot spray if you want to, but it's much better to put it out just as a spray all the way across the paddock. And so in the end, you have lots of bare spots, don't you? And an absolute mess, and all these weedy species coming up. And so people come and do their workshops at our place, and they say, we didn't have any problems finding you because your place is green, everywhere else is yellow. The grass is yellow, the weeds are yellow, your place is green, what's happening? And I'm saying, compost tea. You've got to be joking. I said, it's compost tea. It is good microbial action. But Alan's a little bit naughty. When he puts the compost tea out, you can't actually see in the photograph, but just about half a meter inside this next door neighbor's paddock, there's a piece of area that he sprays deliberately, and it actually doesn't have any capeweed in it. and for some reason or other, our neighbor has not actually ever looked and said, well, why is that? So we have a model farm here, and when the farmers come from the workshops and they say, oh, well, where can we look at a farm that's not as good as this one? I said, well, just look across the fence. You don't have to go anywhere. And so the microbial activity is just amazing. Our stock are healthy. We top the markets when we send them to market. They're cared for and they're respected while they're here. And what we see is farmers coming and saying, well, what do you do for lice on your stock? We don't have lice. Well, what do you do for scours when the clovers come through? We don't have scours. Well, why is that? I said, because they're eating healthy grass. And then the farmers will say, oh, well, we've got our cows, we've got grass up to their bellies. Lots and lots of grass. We've just put urea on. Why? Why would you bother? Why do they need grass up to their bellies? What they need is grass that is highly nutritious, that has the bricks of 10, 11, 12. I said, what's the bricks of your soil, of your grass? Oh, it must be good because there's plenty of it. I said, okay, bring it along tomorrow. So they bring it along tomorrow. And if we're lucky to get a reading, it's about 2, maybe 3. And so I said, you go out and pick some of our grass. Depending on the time of the day, 8, 10, 12. Nutrition is not volume. Nutrition is quality, density, nutrient, grass, if you're talking about stock. It's the same thing with the vegetables. The vegetables will not have pests and diseases on them if you have a nutrient level, a sugar level that is up around 10, 11, 12. You're not going to have that in a piece of land or a vegetable garden if it's full of superphosphate and urea. I mean, to me, it is so basic. It is doing your village farming with a little bit of science around it. Actually looking and seeing what's in your soil, understanding what's in your soil, respecting what's in your soil. And that's what people hear in this panel. This is what we do. We respect the soil and we understand it and we protect it.
Beautiful. Thank you. And what a brilliant introduction for Dr. Lane Ingham to tell us more about what's in the soil. Dr. Lane, I pass it off to you.
Thank you very much, Mary and Peter, for that introduction. That's great. It has been a fairly long, slow road to hoe, getting to where we've gotten to today. Because like Mary, like Peter, when I was first starting my graduate career at Colorado State University to do my PhD, I had switched from marine microbiology, where I'd been working on the microbiome of oysters and what do you have to have in their digestive systems to make them grow? And surprise, it's biology. It's bacteria and fungi and protozoa and nematodes. Without those things, the oyster won't grow. So, places where we have toxic chemicals, outfalls from rivers and streams and such, and that toxic material is getting into the oyster beds, the oysters won't grow. And so, the oyster beds have gone the wayside in Galveston Bay, for example, in Texas. There are only 2 places in the world I will eat oysters from, and that's from Nelson's Bay in New Zealand, where I know that that's clear, clean water, as opposed to up along the coast in Alaska. That's another place where it's good, clean water. Otherwise, I don't dare trust it. And yet, you know, people consume oysters all the time without really thinking about what are they eating? What are they actually getting a dose of? So, you know, trying to get people to understand that we have to do something about this is critically important. When I started doing my PhD at Colorado State University, my major professor, Dr. Klein, said I should go around to all of the people in the science world, in soils. So, you know, horticulture, greenhouse production, out in grasslands, in the orchards, go around to all those people who have any relationship to agriculture, to soil science, and talk to them about the research that you're thinking about doing, which was to try to find a way to measure active fungal biomass in soil without disturbing the system, without having to, you know, indirectly measure what those fungi were doing. And, you know, how do you differentiate fungal activity from bacterial activity, or from protozoan activity or nematode activity? So went out to all of these professors at Colorado State University and explained this is the project that I wanted to do. I was looking at different kinds of stains, doing microscopy, correlating with accepted activity measures like the amount of CO2 being produced, or the amount of an enzyme in the fungal hypha, and presented that information and to a man. And I do have to point out to you that it was only men I was talking to at that time. You know, women professors were, you know, long and far between. So all of them looked at me straight in the eye and said, you shouldn't do that. When you finish your PhD, you have to go find a job. And if your research was on this part of the soil, you aren't going to have anything exciting. You're not going to be able to impress anybody, and you've got to do that in order to get a job. When we look at life in the soil, organisms in the soil, they're just there. They don't do anything. They don't help your plants. They don't do anything important. So you need to come up with a different, project for your PhD. Well, you know, I've caused trouble all my life. And I just went, no, this, if all these people in soils and plant production happily, you know, growing plants in toxic materials, how can you grow a healthy plant if you're covering it with toxic materials 2 or 3 or more times through the growing season? How can that produce be good for you? And so went ahead and started working on the soil food web. And what you see in this picture in front of you is that food web that we put together at the Natural Resource Ecology Lab at Colorado State University. My major professor, Donald Klein, and working also then with Dr. David Coleman, putting together this design where, you know, sunlight energy is what provides for all of this. You've got to have photosynthesis. You know, if you want to talk about magic in this world, there you go, photosynthesis. Taking the energy from sunlight and the carbon dioxides. So the oxygen's going to be blown off. That's something the plant doesn't need that high a concentration of. So out into the atmosphere it's going to go. But the carbon is bound together with another carbon, starting to make the carbon backbone, which is the basis, the backbone of all organic matter, of all organisms, of all the plants. We're all stored sunlight energy in the carbon-carbon bonds within our bodies. So looking at the above ground part of your plant, you're only getting two things from above ground. You're only getting the energy from the sun, and you're getting the carbon backbone, which is that those bonds are storing that energy. Plants can't grow on carbohydrates and sugars alone. You've got to have all the other nutrients, nitrogen and phosphorus and sulfur and magnesium, calcium, sodium, potassium, iron, zinc, chlorides. All of those things make up organic matter. So now where is that plant going to get those nutrients? Well,
but
it all
Elaine,
comes.
Actually, this is a perfect moment before you reveal the great answer. We're getting a bunch of questions about what is compost tea, because Dr. Mary Cole just showed us what it can do, and now, what is compost tea, Elaine? And who is it that's providing those nutrients that you're about to release?
It's the organisms. It's so here we have the whole food web, where you've got to have the bacteria. They're like the base of that food web. You've got to have fungi, especially the more complex the plant is, the later in succession that plant is. What we find is early successional species require mostly bacteria, but they've got to have a little bit of fungi. Things don't grow too well on bacteria alone. With the help of bacteria alone, you've got to have the fungi. And so that component, the fungal component of the soil, increases in fungal biomass the further along in succession that you get. So succession starts with bare soil, nothing's growing. Along comes a few seeds, a little bit of biology in the soil, mostly bacteria, maybe some protozoa, get a little bit of nutrient cycling going, and now you start growing weeds. Well, some people would call some of those weeds herbs or forbs or some other desirable form of very early successional plants. As we get more complex, we start having more cellulose being made by those plants. And so now we've got food in the soil to start growing fungi. So you start to increase that fungal component in the soil. You start to get more and more species of those fungi that help and promote the early successional grasses, the early successional vegetables, the cole, the kale, those early successional plants like cauliflower and broccoli. I love it when my brain starts forgetting words. This is what happens as you get older. You just have to enjoy it. So as we get more and more complex kinds of recalcitrant carbon compounds being put back into the soil by these new and different kinds of plants growing, the fungi become more important in breaking down and decomposing that material. As you keep going, you build up the bacterial predators, you build up the fungal predators, and we're going to have a more and more complex food web, more and more complex set of nutrients being cycled and made available to plants that need higher and higher quantities. So think about the amount of nutrients that an old growth forest tree needs every single year. And that plant is going to be sequestering away in its bark, in its woody layers, the rings that it puts on every year. And so every year that plant requires more and more of all of these nutrients, and you're taking them out of circulation. So where are those nutrients coming from to keep that tree alive next year, or 100 years from now, or 500 or 1,000 or 3,000 years? Where are those nutrients going to come from? And nobody's out there putting on inorganic fertilizer. Nobody's out there putting out the phosphate or the sulfate that the plant is going to require. So what's the trick? How did nature manage to do this, maintaining this massive biomass that has to be produced each year? Where do the nutrients come from? And it's got all about to do with the bacteria and the fungi and the protozoa and the nematodes. So bacteria and fungi are grown. When you get into these systems, the plant is going to put out exudates, which are mostly sugars. a little bit of carbohydrate, a little bit of protein, amino acids, for example. And those exudates are specific for the bacterial and fungal species to get them to grow and make the enzymes that are going to pull the nutrients out of the inorganic material, the sand, the silt, clay, the rocks, the pebbles, the parent material. Every second of every day, your parent material is breaking down. A process done by the bacteria and fungi and their enzymes and their acids that they produce. And so every day that nitrogen, phosphorus, sulfur, magnesium, calcium, sodium, potassium, iron, zinc, all those nutrients are being replenished in your soil. You will never run out of those nutrients in your soil as long as you have a good, healthy set of microorganisms that will be able to pull those nutrients out of the sand, the silt, the clay, the rocks, the pebbles, and oh yes, the organic matter as well, pulls those nutrients out, stores them in their bodies, and then the protozoa and the nematodes, the predators in the system, come along and eat those bacteria and fungi, releasing those nutrients in a plant available form. So that's how that plant is always going to be healthy. Well, let's go backwards along succession. Does that happen with your corn? Does that happen with wheat? Does it happen with barley? Are these same processes going on when you're trying to grow onions and earlier successional vegetables, tomatoes, potatoes, a little bit later successional? So all the way back to those weeds that people so abhor. But every bit of this planet has to go through that stage of being converted from dirt, where you have no organisms present, or you only have some of the less desirable bacterial species present. They have to make way for these ever more beneficial sets of organisms, a more diverse sets of organisms in that soil that do the nutrient cycling, that gives the plant all the nutrients that it needs. And remember that the water that runs from uphill, downhill, every time any of that leaching starts to happen, as those leachates run through your dirt, hopefully some of your soil, those nutrients are going to be pulled out and put back to replace the nutrients that your plants removed from that soil. The only way to make those nutrients available to your plant is to have them processed by the bacteria and fungi, the protozoa, nematodes, microarthropods, earthworms, all the things that eat bacteria and fungi, so that those nutrients will be released into the root system of your plants in exactly the balances that the plant requires. So you can stop worrying about how much nitrate do I have to put in? How much phosphate do I have to put in? And what about all the other micronutrients? Why don't we ever hear about you've got to put in all the micronutrients because the plants require all of those nutrients, not just nitrogen or phosphorus or sulfur or calcium or magnesium or whatever. You've got to have all of them in order to grow a good, healthy plant. And the only way to do that is to have a healthy set of microorganisms that the plant will wake up with its exudates whenever the plant needs. It's not getting enough. It's going to put out those signals to the bacteria and fungi. I kind of like to think about that as being like the pizza delivery version of what we do in soil. So the plant picks up the telephone and calls the exudate producing part of its body to make the specific order. And so the pizza delivery guys will come by, pick up the order. They go and make the pizza. Now, these are the bacteria and fungi working for you here. They make the pizza. They put it all into the box. And then here comes the delivery boys, otherwise known as protozoa, nematodes, microarthropods, earthworms, and such. And they're going to deliver to the surface of that root system the nutrients that the plant requires in the proper balances. And how can you do this? If you're killing the very things that will give you fertility, that is giving you the healthy food that you need, you've got to have these microorganisms in your digestive system as well. We can't forget about the fact that all of those organisms that are in our digestive system come from the surfaces of the plants that you eat. You don't want to be washing them off. You don't want to be treating it all with nitrous oxide. You're killing all those really beneficial organisms off. So how do you make certain that there are no pathogens coming from what you're growing in your garden or out in a pasture or out in a agricultural field? You can't be putting out toxic chemicals that kill the good guys and thus prevent your plant from being able to resist the diseases and the pests and the problem organisms. So we have to have this whole food web in the soil at all times, and we've got to learn how we promote it. How do we make it always be present and always be functioning so you and the animals that you're feeding, your family, require without the threats of those of toxic chemicals or those diseases and pests that you don't want to be catching. So we try to introduce people to the whole food web there. And I know it generates a lot of questions, which is why we have the Soil Food Web School. And so the foundation courses give you the introduction to all of the things I'm just talking about, and a lot more detail on exactly how does a successional thing work and exactly how is it that the plant knows what nutrients it needs and how's it going to put that information out, and all of those steps through very not complex.
And we have a great time reinstating it. It's wonderful. And so at this point, one of my favorite things to do is to bring folks who think about similar organisms and give each of you a chance to learn from one another as well. So I'm curious, Peter and Mary, or Elaine, do either of you any of you have a question that you'd like to ask in this room that maybe we could discuss together? Right
Go
on
ahead,
the
Mary.
spot for you.
Yeah, put us all on the spot.
No, well, I just find it interesting, and it's because I have two interests. One is the mushrooms, and I'd like to talk to Peter about that because it's very interesting here in Australia. With mushrooms, a lot of introduced species, and as a person with the Poisons Bureau, we always were able to look at mushrooms, for example, Amanita phalloides, as being attached to introduced species of oaks, etc. What we're now finding, both in around Melbourne, Canberra, and in Tasmania, is that Amanita phalloides in Melbourne and Canberra is shifting host to the eucalyptus species. And in Tasmania, we're finding that Amanita muscaria is moving into the native vegetation. And so I find this an interesting situation that's happening, and it's happened within 200 years. And this is very rapid in terms of ecological evolution. So I guess my question at a mushroom level is, do you see this happening within the USA or whatever, that we're actually getting these host shifts for what are in effect mycorrhizal fungi?
I mean, I hadn't heard about that in Australia, and yeah, that's definitely a bit alarming. I've heard of something similar in California. I know that Amanita phalloides. I don't know the details. I know that it's been a bit of, as far as I understand it, a volunteer effort, I think, with the Santa Cruz Mycological Society. I know Christian Schwarz, who's a well-known and respected mycologist, wrote a great identification guide a few years ago with another author. for like California mushrooms. That club and maybe one of the universities in affiliation, I'm not quite positive, but I imagine, would hope, has been looking at the same thing with Amanita phalloides being introduced from European oaks is the hypothesis, and seeming to replace the native Amanitas. I'm not sure if they've concluded the research or where it's currently at, but I know that that's definitely a concern. For the audience who doesn't know, Amanita phalloides is one of the it's one of the deadly toxic mushrooms. There's not very many in the world, and it's one of the more notable ones because a lot of people misidentify it, especially in North America and probably in Australia, I imagine.
Australia
too.
as well.
Yeah, a lot of immigrants, usually from parts of Asia, where there's a very similar looking edible mushroom. And so they'll come here and eat that, misidentify it, and their liver shuts down in a couple days. So it's very concerning. And especially just, the shifting in ecology and the sort of pushing out and sort of the ecological strength of this toxic mushroom. It's kind of, what does that even mean? I don't know. But I'm not aware of other shifts. I mean, this is one of the massive questions in macrofungal distribution, which is why in North America, there's a nonprofit that was started a few years ago, originally named the North American Mycoflora Project, and now they're called the Fungal Diversity Survey, I believe is their new name. But essentially, it's a largely volunteer effort, either individuals or mycological societies getting internally funding or applying for grant funding for genetic sequencing of species that are collected, macrofungal mushroom species collected in 1, 2, 3 day myco blitzes, to start to build out distribution maps of the fungi, macrofungi of North America, because we basically know next to nothing. Anytime you travel across the country, you get the local field guide, you find a species that's not supposed to be there, that's there. We don't know how things are drifting around. And unlike all the plants, and especially invasive plants of North America, which are very well mapped for their distribution, you have next to nothing. So it's one of the biggest arguments, or one of the not arguments, but one of the most accessible ways for the amateur mycologist, amateur meaning the lover of fungi, to actually contribute heavily to the citizen science, citizen mycologist arena.
Absolutely.
This important field is helping us just document where and when fungi are found. So it's not just distribution patterns and maps, but also the fruiting habits and the timing, and how is that shifting as climate and temperature windows shift and things like this. This is a huge question. How does that impacting degradation cycles, nutrient cycles, as different decomposers maybe are fruiting in different times, and how that impacts the insects and everything that eats off of the macrofungi. And all of that is only going to be collected with individual data points, which you cannot predict, unlike plants that are there every year. Mushrooms, you have to go out, find them in a narrow window, and then ideally nowadays upload it to websites like iNaturalist, or Mushroom Observer, which is specific for fungi, or through the Fungal Diversity Survey if you're in North America, which works with both of these projects, both these websites, and a couple other. It's like a multi-pronged thing to document the collection and where and when it's found through iNaturalist and Mushroom Observer, collect the species, send it to any number of universities to get sequenced, upload that data to name escapes me, but the global genetics database, and then ideally as well send the dried collection to an institutional fungarium for preservation. So we have sort of everything's well documented, and it can always be maybe resequenced or used later for any other research with the actual physical specimen. So that's, there's a whole step-by-step protocol through the Fungal Diversity Project's website, and that's, anybody and everybody can and should get involved in that, large or small ways, because it's only through one collection, one documentation at a time that we're slowly going to build these maps and understand what's shifting, what's moving where. And yeah, of course, especially with invasives, actually just popped in my head. You were asking about mycorrhizal, maybe a more compelling or alarming example of an invasive mushroom is with the yellow oyster. So shiitake mushrooms have been cultivated in North America since the 70s or so. They were actually, as I understand it, banned from importation and cultivation because there was concern that they would become invasive, especially on train tracks, because there's a mushroom called the train wrecker that's distantly or somewhat related to shiitake that can be known to grow on railroad ties, degrade them, and has historically caused derailments. So shiitake was actually banned from importation for a long time until it was determined that they wouldn't do that. Surprisingly, after all these decades, even though I've heard rumor, I've never seen any documented, publication of actual shiitake jumping from an oak log in your backyard, your property, into the oak forest right next door. For whatever reason, they don't seem to be established. And that's a big question. There are ecological questions why that might be, especially when we have, similar climates to Japan and parts of North America. But the yellow oyster was only introduced about 10 years ago, and it's a subtropical species. Primarily is where it's found. It's quick growing, easy to grow, eats all kinds of stuff from a gourmet mushroom grower standpoint. And it seems that based on sort of preliminary research from Andy Bruce, a mycologist who went to one of the universities in Wisconsin, basically she was noticing on her runs in the forest in Wisconsin, yellow oysters, and started asking her professors and friends, like, hey, this isn't native. This is like a subtropical mushroom. Why is it way up in Wisconsin, almost to Canada? And she starts putting out calls on Reddit and asking other people on Facebook and stuff, seeing if other people were seeing yellow oysters, in their forests, and just was getting flooded with photos from 5 years prior, New York, upstate New York. And so it seems that this mushroom has just been took off, just super aggressive, as oyster mushrooms are, really easy to grow. And especially the yellow and pink oysters, which any cultivator knows are really quick growing, really easy to grow. And that seems to be like maybe our most clearly, invasive, if you will, saprotrophic mushroom. Huge questions on what it's doing to replace native oyster species, other decomposers, etc.
Yeah. We cultivate them here in Australia, and in Australia we have the fungi map out of the herbarium in Melbourne for anyone who wants for citizen science. But it's the mycorrhizae that I'm particularly interested in, because it's the mycorrhizal fungi that are destroyed so quickly with the synthetic, particularly with superphosphates that are used here. And so it's the mycorrhizal fungal mass, and this is where, coming back to Elaine, the mycorrhizal fungal mass is just so important if we're going to be looking at this increased sequestering, well, not only sequestering of carbon, but actually holding it there. It's not too difficult to sequester carbon, but it's very difficult to get it to stay. And the only way that we're going to get a good fungal biomass back into the soil again is to stop this cultivation of the soil. And Elaine, it's, if we continue to cultivate in conventional agriculture, we're never going to increase our fungal biomass like you and I know we can have, which will help to give us particularly into climate change, good soil quality, good soil moisture holding capacity, good carbon capture, good health, good resilience. What are we going to do if we're going to continue this conventional agricultural system?
We can't. That's the bottom line. We cannot continue with the green revolution.
Yes.
I imagine someday Niels Bohr, who started the green revolution, is, instead of being hailed as a hero, is going to be hailed as the doom of civilization, because it's like reading David Montgomery's book about what killed ancient cultures. Well, every single time it was destruction of the soil. And what's really lost are the microorganisms. It's not like the sand or silt or clay or even the organic matter goes away. They can all be there, but it's no biology to do decomposition, to cycle nutrients, to protect the plant roots, doing all of the carbon sequestration back in the soil. So it's critical that we understand those things and now start growing crops in a very different way. We can't till. And so how do you keep the soil nice and fluffy and loamy, lots of good organic matter in there? Something has to build structure. Well, who's the something? It's called fungi and bacteria. Bacteria start the process because they glue together the micro aggregates. But that's not enough to give you good inflow of water or oxygen or even your roots. You've got to have the fungi come along and bind things together. And now you've got all this space in soil to store water. There's lots of oxygen coming through, so you're never going to have the situation where your roots are going to be killed by the media going anaerobic. Roots are obligate aerobes. They cannot tolerate anaerobic conditions. And therefore, as soon as you've got anaerobic conditions starting, well, and what starts the anaerobic condition? It's compaction of the soil. You have to protect the soil surface. When you think about raindrops coming down and smacking onto the surface of the soil, those are compacting effects. Think about when a big drop of water hits your head. It can be a not very pleasant thing happening. Bam, 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 bam. And we run for the house. Well, the roots, your soil, the roots of your plants can't run to the house and get protected. We have to have understory plants that the energy of that raindrop is going to fall on the surface of that leaf. And then when it hits the soil, it's not going to be compacting anything at all. We've got to stop tilling, because every tilling machine pushes down on the soil where the tines push wherever they go down to. Now there's where your compaction layer is. So we can't allow either of those things. We've got to protect the surface by putting a nice thick mulch layer out, except that when you're in a productive system, that mulch is going to decompose in a couple of weeks. And now you got to go out and put on more. I am not interested at all in having to put on 5 or 6 applications of mulch on the surface of my growing. If I've got 10,000 hectares, I can't manage to understand.
So I'm popping up a question here that was related, because there's this notion that tilling disrupts the fungi, but it also, as you're describing, Elaine, it does more than just disrupt the fungal structure, and which then compacts the soil. It then perpetuates those compacted conditions, which makes it harder for aerobic organisms to thrive. So we had someone ask, Armand asked, I appreciate how no-till avoids damaging fungi, but what about a scuffle hoe? How deep can I go to remove weeds without damaging fungi? So I'm wondering from you and with Mary, just different techniques, because we have a few other questions about how different techniques in working with the soil actually affects the fungi. And what's your experience with that?
Go ahead, Mary.
Oh, well, first of all, there's weeds. What are you calling weeds? Weeds to me are mulch potential. I don't see weeds as a problem. They're primarily colonizers. We've had this situation here in Australia where we've had, like in California, we've had devastating fires. And here, just only very close to us, we were close to fires in 2019, I think it was. And there was a lot of destruction. And so then when the farmers were then looking at their paddocks, they were having what they were calling weeds coming back. And the departments of agriculture were saying, oh, well, you need to get out with your glyphosate and get rid of them. And I'm saying, no, please stop. Please let me talk to the farmers about what to do. The weeds are there. They are part of the seed bank. They are what you call weedy species. They're primary colonizers. They're starting the nutritional going again. Some of the fires were very hot, so there was a lot of destruction. And so I'm saying, just don't let the weeds flower. Don't get rid of them. Don't let the weeds flower, and put out your fish and kelp and molasses and compost tea, and actually start to stimulate the microbiology so that you change your soil. And Elaine can talk about it. Change the soil so that your weedy species are not coming through as the predominant species, that your perennials are starting to come through. So just don't get rid of the weeds and leave bare soil, because not only, as Elaine says, the droplets of water hit and compact, they also run off. They cause erosion. You're losing your topsoil. So the weedy species actually are very important. And so as the primary colonizers there, so when you're saying, how do I remove the weeds? Well, if you had good soil microbiology, you wouldn't have so many of those weedy species. So it's start farming the soil and improving the quality of, or the ratio of your bacteria to your fungi, and those weedy species won't be coming through. And what we do here in Australia, I do a lot of work with the wine industry. And what we do is we simply use if we're going to do anything at all, we simply use a crimper, a sort of a machine that will knock down these plant species, and then they become the primary source of organic matter for the bacteria and the fungi, particularly for the bacteria. We just don't let them flower. We don't get rid of them. We just don't let them flower, because what they're doing is they're holding the soil together, and they're starting the primary nutrition, the primary nutrient conversion that whatever the permanent plant is going to use in the future. So I don't see weeds as a problem. I see them as primary colonizers. I see them as mulch potential. I see them as absolutely essential when you've got like a fire situation, or you've got a bare earth situation for whatever reason. And so don't remove the weeds, work with them, manage them.
I know Dr. Suzanne Simard also talked about the potential for those intercropped plants to maintain mycorrhizal connections as well for year-round mycorrhizal networks, which ties in nicely to Nancy's question. And I think I'm going to pass this ball over to Peter.
Can I take a second to finish a question that I was asked previously? What is compost tea?
Oh, yes.
And
what is compost tea?
Got into trying to define compost first. And so what we want to do is grow all these great, good fungi. There's lots of different kinds of strands and colors in here. You can see a lot of the bacteria, a couple hundred species of bacteria, more or less in this picture, some protozoa. So we want all of these organisms growing in the compost. We have a minimum value that we want to get to, regardless of what plant you're going to be growing. You want at least this level of these organisms, start getting them put out onto the land. And a lot of times putting a solid out is very difficult to do. So a solid compost is more than a lot of people want to do. So what you do is take your compost, put it into a container, and make certain that you start moving the water up and through your compost, and you're extracting all of these organisms from the surfaces of the organic matter, the sands, the silts, the clays, things like that, that might be in this, in the compost. And so all these organisms get out into the water. Typically with a compost tea, because what I just described was a compost extract. But if you're going to do a tea, then you're going to add foods to grow the organisms that are missing in your soil. We want to get the fungi up to a certain level. Maybe we want a fungal to bacterial biomass ratio of 1:1 or 5:1. So what we have to do perhaps is grow a lot more fungi. So we put fungal foods in and we let it brew for about 24 to 48 hours, depending on temperature. And then you just take this liquid and you use your sprayers and you spray out on the above ground part of your plants, and maybe a drench around the root systems as you go by. And so you put all these beneficial organisms back into your system. So an easy way to get these applied, especially if you have overhead irrigators, or you've got different kinds of arrangements where you put irrigation water out. Well, you can put the compost tea as well.
Mm-hmm.
It's important. Can I just follow on, Elaine? It's important that that the farmers take the filters out of their systems.
Oh yes.
and because the one thing that gets clogged up, my filters are all clogged up. Yes, well, of course they are. It's all this beautiful fungi that you've been trying to put out that you've now got all over your filters. So take the filters out, and don't have the pressure of the system too high, because it will kill the microbes. But it's absolutely a really good way as long as you take the filters out of the system.
Yep. Yep. It's a good way to get a lot of the spores too spread
Yeah.
through your
Yes.
whole system. So
Absolutely.
what we try to do is get a lot of perennial plants growing. So then those plant root systems are colonized with the mycorrhizal fungi. So the
That's right.
next crop that you put into the furrow, you're not tilling out here away from the furrow. You're just tilling the furrow. You drop your
Yes.
seed with some of the compost tea or compost extract that has the spores of the mycorrhizal fungi in it.
Yeah.
And so, you're going to add those perhaps as, go get your gills, wash off the gills in a little bit of water. And now you've got your mycorrhizal spores in your tea as well. You put them in last thing and go ahead and spray. And especially if you put it in furrow right before the seed drops on top, you're going to get very rapid colonization, both from the existing perennial plants that will colonize really rapidly, as well as any place that doesn't have really good mycorrhizal fungi left in the soil. The spores will germinate and grow and colonize that root system.
Mm-hmm.
So Peter, does that...
And just for, so that everyone knows that we're on the same page at the same time, mycorrhizal fungi are typically the ones that associate directly with living roots and either are arbuscular and get inside those endomycorrhizal or the ectomycorrhizal that can form these nets around the root tips. But actually, Elaine, that was a perfect segue to this question, but I'm going to start with Peter. Will you please talk about the different ways that saprophytic and mycorrhizal fungi decompose organic matter and minerals?
So there's a little bit of a spectrum in my mind of fungal and ecological niches, and some species kind of can switch between roles. So we might pigeonhole a given species to be primarily saprotrophic, saprophytic, or parasitic, depending on the day. And some seem to also can be somewhat mycorrhizal, and even some species can have different types of mycorrhizal relationships. So it gets a little nuanced, of course, as the sciences tend to. The standard definitions, the sort of introductory distinctions, is that your classic pure decomposer mushroom, something growing on a piece of wood above ground. Well, yeah, we can start with, wood degradation is fairly complex, but the wood decomposers are releasing, depending on the type of wood decomposition they perform, different suites of enzymes and often hydrogen peroxide as a means to catalyst the oxidation of lignin. And it's a slow burn, cold burn, as we call it, to break down wood. It's really complex without getting too much in the details just yet, unless we want to go there, because it's really important for soil building is to break down of wood. But they're basically eating stuff and they're breaking it down. They're digesting externally. They're releasing dynamically what they need to release at the tips of their mycelial network, the right compounds, digestive enzymes, primarily for organic matter, to break it into peptides, amino acids, simple carbohydrates, and then those byproducts are taken up through absorption, primarily through the tips of the hyphae, and then there's a lot of byproducts that are left for the rest of the organisms. So that's a simple way to think about the saprotrophs. Saprophytic, just to nuance that even more, so it's if it's phytic, it's breaking down plant material, but if it's saprotrophic, it's eating all kinds of stuff, not just plant material, because fungi grow on all kinds of stuff depending on the species. And then mycorrhizal fungi, there's 7 categories that are officially recognized, and they're distinguished primarily by the structure they form, the means of exchanging nutrients between them and their plant partner. And we've primarily focused energy and research into the ectomycorrhizae that associate with about 5% of trees or plant species, mostly trees and shrubs. And then the arbuscular mycorrhizal species, which are one type of endomycorrhizal species, because they're found in roughly 90+% of plants, or can associate with 90+% of plants, and are super critical for ecological function, arguably the most ecologically significant of all fungal groups. And then they have a huge potential in agriculture and food production, plant production. So, and the way that they break down food depends on this sort of how they access nutrients, rather depends on the type of mycorrhiza. So broadly speaking, we think that we talk about the arbuscular mycorrhizal fungi aren't so good at acquiring carbon and nitrogen from the soil, but are, most notable rather for their access of phosphorus for their plant partner. They don't really get so much carbon. That's what they're getting from the plant partner. So they're releasing not an enzyme, but here organic acids that help solubilize, sort of make water soluble, make it available, shift the energetic state of phosphorus from mineral forms, rocks and things, and take that in, transfer it over to their plant partner, and then get that photosynthesized goodness in exchange. The ectomycorrhizal mushrooms and truffles are a bit different. They don't do that quite as much. They're more notable for providing nitrogen to their plant partners, and they actually seem to. And last I was digging into this, maybe Mary, you have, or either of you have more updates, but they surprisingly seem to degrade some actual food in the ground using enzymes, organic matter rather, but seem to only mostly get the nitrogen out of that, but don't acquire the carbon. Sort of, we don't, last I heard, don't know why. So they can actually access a good bit of nitrogen from the soil, even from springtails, little small insects. So they can actually be kind of like carnivorous in a way, which is pretty interesting. Yeah, so different niches performing different functions. And then, as was briefly mentioned, if folks aren't familiar with Susan Simard, a really well-known and respected mycologist, done a lot of research on ectomycorrhizal fungi and has a good number of TED Talks and things. Her work has looked at how these fungi are not only accessing nutrients, but spreading them throughout an intact ecosystem, especially a dense forest, and moving them between plant species and where preferences are made. Whereas the saprotrophs, they're just eating what they're growing on, and then stuff falls away and other people benefit sort of by happenstance.
There was an interesting question about uptake of heavy metals, things like that, by mushrooms. What's really interesting, if you remember back when Chernobyl blew up, the cloud of radioactivity was actually monitored across Scandinavia and down into Europe by harvesting mushrooms and testing the amount of radioactivity that was picked up in the mushrooms. The other thing that you may or may not remember, there used to be lead in petrol. And it was always a problem that one didn't collect mushrooms from along the roadsides when they were using leaded petrol. And I remember my father saying as a child, we don't pick the mushrooms from along the side of the road because they pick up the heavy metals. So in terms of the mushroom species, yes, they do pick up heavy metals, and they're actually used as indicator plants when there has been any sort of pollution. So it is problematic in terms of how one or where one collects from and how one uses them. And also there was a question about hemp. Hemp also takes up heavy metals. So it's going to be interesting in the hemp industry as to how we measure and use those as food resources. And that's something that I'm involved in at the moment.
Well, and we had another question come in about glyphosate as well. Are mushrooms and other fungal components, mycelium, are those fungal tissues accumulating things like glyphosate?
That's an answer I don't actually know, because I haven't actually analyzed, but I'm interested in, I know that here in Australia, where glyphosate is used, we're seeing an enormous drop back or reduction in the amount of not only saprophytic fungi, but mycorrhizal fungal biomass in an area under where glyphosate has been used, even though the manufacturers are saying that it's decomposed very rapidly. We have taken soil directly under where like a broadleaf weed has been killed, and we're finding, if we look at another area
There's been a fair amount of work done by Dr. Don Huber, who was a USDA extension person, and he's got many papers on the effect of glyphosate at this point. He waited until he finished his stint at the USDA because he knew that if he wrote papers that were negative to glyphosate, he would have been, dropped. He would have been fired. It would have been something else was made up to get rid of him. But he knew that he couldn't publish any of that until he he retired from the USDA. And Don is amazing to listen to and his experiences with what glyphosate actually does. Glyphosate is a pretty good sterilant in most instances. If you have those bacterial and fungal species that can decompose glyphosate, then you will see the 2-week time period for decomposition and the glyphosate is gone. So if you have the right microorganisms, then it acts as Monsanto said it would act. But if you've killed off those microorganisms by putting out inorganic fertilizers, pesticides, herbicides, et cetera, whatever, and those particular species are gone, then you're not going to decompose any of that glyphosate and it will accumulate. And there is some evidence that the glyphosate will start getting into the plant and move up into the seeds and the, what we as human beings, what we eat is going to have elevated glyphosate concentrations. And so Gabe Brown and Don Huber would probably be my 2 recommendations to get some of their papers and read some of the research that they've done.
Well, and Peter, it's worth coming back to, I know you have a branch of your thought is in mycocycle or in recycling via fungi. Am I thinking correctly?
Yeah, I'm no longer affiliated with Mycocycle, but I
got
a lot of history with mycoremediation for sure.
Indeed. And in your experience, have you either in cultivating mushrooms had trouble with various materials because of toxic contaminants, or just experienced the power of fungi to decompose those heavy metals or pesticides, etc.?
Yeah, both. So yeah, just like a super quick microremediation crash course, probably a lot of folks might be interested. I like to think of what fungi can do in this regard with based on the 2 different types of contaminants we're often talking about, whether it's a chemical, a molecule, many atoms bonded together in a structure that does a thing, versus elemental heavy metals that maybe, singular atoms or a couple atoms that are found in a common state in nature doing their own thing. Fungi can't transform lead into gold. They can't turn mercury or arsenic into something healthy. They just move that stuff around and maybe bond it with something to stabilize it, make it less impactful, as other organisms can also do, or they accumulate it, as many plants do, into their tissue, and then a reindeer eats it, a deer eats it, then outside Chernobyl, then a human eats that reindeer that has this toxic, radioactive cesium, and this stuff never goes away. So with heavy metals, whether radioactive or not, there's certain things that fungi can do, but they can't make that stuff go away. That's always going to be a problem. These metals are most primarily meant to be in tiny concentrations in the topsoil or deeply embedded in the earth, and never really meant to be concentrated through extraction processes, let alone spilled by accident or in a giant cloud, but poof. So, and then we have the chemicals, and this is where fungi shine with their ability to degrade all kinds of things. The hypothesis using the same oxidation methods or processes for breaking down wood, applying that to a lot of really recalcitrant compounds that are complex, strongly bonded, and resist normal enzymatic decomposition by, say, bacteria. And so depending on the compound, depending on the fungus, depending on the pH, the temperature, other ambient conditions, whether nutrients to fuel the fire are available for the fungus to get the ball rolling, you're going to have different effects. And this is what a lot of the history of microremediation has looked at, and really where the highlighter comes out is, you know, in this study, it's that these combinations of variables gave us this percentage of degradation in the lab. So how do we translate those really specific variables to field conditions, right? Maintain consistency with a living organism. That's where it gets difficult. So you have a better chance in getting results of breaking down toxins, really being wowing others and yourself with doing something in the realm of microremediation under controlled conditions, at least to start. That's where we're at these days. And that's going to be easier depending on the fungus. Some are powerhouse, some strains, some varietals of a given species are better than others, just as we find with plants that do same species, different varietals of the same plant will perform different under different conditions. And then pairing that with a given compound. So that's the quick crash course. To answer your question, you know, both. So, you know, the way to go about this as the garage microremediator is just start to throw a fungal strain, some mycelium on a toxin, you know, do it safely, of course, and hopefully with good experimental design and record keeping. But you can do this. I often do little, you know, quart jars or early pint-sized jars, small volumes, different ratios of whatever the waste stream is, prepare it different ways. How much do I hydrate it? Do I cook it? Do I not? Do I mix in other supplemental ingredients to, again, provide supplemental nutrition to the fungus? You know, mix that up to the end of days, and then throw in your inoculant and just see what happens. And it's really basically rote fungal cultivation. When you boil it down, if anybody out there is a grower, sounds maybe familiar to you, and you're just waiting and seeing. And that's what a lot of the history of cultivation, mushroom cultivation especially, has been for the last 100 years, trial and error by lots of people. And now for the last few decades with microremediation, you know, in a more refined and, you know, measured way with compound concentrations. So we can pretty easily replicate that in lab. If you don't have analytical gear or can't afford testing, you can definitely observe, does it grow on it or not? It's a really basic thing. In my book, I have a photo actually where I sterilized some pieces of paper, dipped them in glyphosate, threw that in a petri dish with, you know, 10 different strains or species of mushrooms. You know, the enoki just went right around it, did not want to touch it. The oyster went right over it. You know, it's pretty easy to do yourself. Yeah, so it's hopefully that kind of answers and goes not too far off track. Kind of coming back to, well, I guess I'll leave it there for now.
And there's one other thing that I'd like to say that I think it's a general comment, but I think it's important. When we're talking about the soil and we look at research that's done on the soil and different outcomes, I think what's really important to think about is that what is the soil? What was the soil that was being used for the experiments? Was it a soil that has been over-cultivated, has had lots of conventional synthetic chemistry put in it over a very long period of time, and the reaction that happens there, versus a soil that, you know, we here are all wanting to see, where it doesn't get cultivated, where it has 100% grass cover or ground cover all of the time, where it has a highly diverse microbial population. If you do research on those sorts of soils, the results you're going to see in a lot of the academic papers and a lot of the technical papers is going to be very, very different from what you see with where most of the research is done, particularly by the chemical companies, is on soil that's not soil at all, it's dirt. And so I think it's important that we really have a think when we're looking at a paper, was this research done on dirt, i.e. conventional agriculture, or was it done on soil? And I would put it to you that most of the work has been done on dirt. So I think it's important, just as a general statement, that we really think very carefully when we're looking at results. What are we talking about? Are we talking about the soils that we're all talking about? Or are we talking about destroyed, damaged soils, dirt, over very many years?
Mm-hmm. Soils
'cause-
kicked back to their earliest successional stages.
Yep. Because what we need to know about is where we've got all of the functions of a happy, healthy soil food web, because that's where we've got to move to. We don't
Yes.
really have a choice.
Yes.
And then understanding how that food web shifts and changes across these gradients that we can create to cultivate different crops. So we talk a bit in the Soil Food Web School about succession. And actually, one of my students, Marcus Kirkdyke in Australia, asks, is there a succession within the fungi like there is with plants? And how much do we need to keep this in mind when restoring soils?
The attitude we take is we want to maximize diversity. So we're going to try to get absolutely everything in there. Of the indigenous species, you don't want to be writing to Mary and asking her for all the fungal culture she's got. She wouldn't send it to you. But if you find someone foolish enough to do that, you could be bringing over the next disease that wipes out the ability to grow corn or peanuts or something like that. So you got to be a little careful to limit it to the indigenous species of organisms that are in your bioregion, so that we're not bringing disease, or you could bring a organism that seemed completely harmless, and then it turns out to be the worst disease-causing organism ever seen in that part of the world. So be careful. Maximum diversity of all of the mycorrhizal fungal spores, and gosh, where would you go to find that? Peter, can you answer that one?
Well, yes. I guess just in addition to just general succession, we do see with decomposers, there's several stages of fungal decomposing fungi when you're like watching a rotting log. So that exists. That's documented and pretty easy to learn up in mycology textbooks. Yeah. And then with ectomycorrhizal mushrooms that are a bit more observable and measurable, I think I've only seen one paper that basically speculated based on their observation of a deforested area and it's coming back to stability, something in the ballpark of 40 years they approximated before the ectomycorrhizal community seemed to return back to what it was, or back to normal, or something more stable. But I mean, these things, fungi work on geological timescales, and we've only been looking for a very short amount of time at this stuff. So it's hard to really say as far as they go. And then as far as what Elaine was just talking about, the below ground fungi, especially how to get the inoculum, is where I can speak to a little bit more. Again, we primarily know much more about just the ectomycorrhizal mushrooms and truffles and the arbuscular mycorrhizal fungi. The other 5 groups of mycorrhizal fungi are just very poorly studied, and the effectiveness of either creating or sourcing or trying to harvest from nature and amplifying indigenous inoculum is just not as well sort of proven because the lack of experiments. But as far as arbuscular mycorrhizal fungi, it's hats go off primarily to the International Vesicular Arbuscular Mycorrhizal Culture Collection Center in VAM at the University of West Virginia University. They have a lot of protocols, methodologies on there talking about how you can harvest soils, indigenous soils, sieve the soil to get these spores out. Their spores are very large and actually can be trapped in standard soil sieves and washed, identified under scope if you want to go that far, and then amplified. There's even more low-tech approaches that the Rodale Institute is, and some of the people there are really well respected for, spending about 7-8 years figuring out very low-tech methods of just harvesting soil. Throw some allium seeds at it, or some quick growing subtropical grass, and without even looking under the scope or sieving, you're just hoping you're going to grow some of the indigenous arbuscular mycorrhizal spores, and then use that soil the next season, and the root tips and everything that's become mycorrhizid, and the spores that are in there as inoculum, or dilute that into your soil mix the following season. So you can look at those sources. They're pretty well known, and most books talk about them as the go-to. And then the ectomycorrhizal fungi, if you're dealing with like a woodlot or shrubs or more perennials, something that's known to need, or you're just guessing or hoping it might associate with an ectomycorrhizal species, you could just go grab any, tree, shrub, associated mushroom in the forest and harvest its spores through a spore print, or putting it in some water and let the spores drop, or a puffball, and just getting those. The ones that are best appreciated that you're going to find on the species listing of most commercial products are the ones that have just been shown through the decades to be most consistent and reliable for associating and or associate with a wide range of plant hosts or plant partners. So a real standout one is Pistillitis Tintorius or Pistillitis oryzus. Dead man's foot doesn't look like much. It is actually edible and has some other ethnomycological applications, but it tolerates high temps, contaminated soils, associates with tons of trees. It's really common around the world, though it might not be fruiting. It's in many soils. So that's a good one. Lacaria mushrooms, where I'm from, are edible and actually medicinal, and strongly associated with some of the timber crops around here. So they're very common. And then a number of other puffball species, Swillis, or not Swillis, but Rhizopogon is a good one. And then
Is there a
Swillis
place
species.
that people could go and buy those?
Well, so if these mushrooms, so I've sort of thrown these genera out at you, if they grow in your area, you learn to identify them. You can, as I just sort of mentioned, and you said earlier, Elaine, you can just harvest the spores. And commercial products, they're probably getting them in the wild and diluting them heavily in bentonite clay, because you don't need a lot of spores, and you got to stretch your product out or they're getting them from the wood lot where they're already growing. They just repeat the cycle. Otherwise, yeah, you're just looking for commercial products. Now, I can't recommend one over the other. What I can say, and I've heard from many people over the years in their mycology class or as a side fun project, they look at commercial mycorrhizal inoculant products under the scope and sieving out the spores and whatnot, and often find the marketed or advertised spore count to be way off the mark. And in some products, I've been told by students, they weren't even able to find any spores in the product. So again, I can't recommend any one over the other, but just do your research, or maybe do some old trial plots and this kind of thing, or make your own, especially with an indigenous, not only indigenous species, but an indigenous strain that's really adapted to the local climate, elevation, soil, etc.
Mm-hmm.
Yeah, I mean, I'm just kind of thinking about people in our audience trying to get some of these inocula to go into their, mix them in the compost after the composting process is finished, not during. And, if there was, who would they call? Who would be people who have this kind of knowledge? So maybe, they could get hold of you on your, on your website and get some ideas on who the people would be that could help them identify and gather all of those indigenous species.
I think this is, along with all the other ways that we test soil, and it's some of the work that I know Mary does, it's especially with the arbuscular mycorrhizal identification and strain collection and amplification, even just harvesting native AM fungi, AM spores, amplifying them, and then providing that as a local, it's sort of paralleling, like you just said, paralleling the locally made compost, really high quality compost tea, fresh stuff at the farmer's market. I also have, the indigenous arbuscular mycorrhizal inoculum that was made from, our local forest or whatnot. I think that as the word gets out about the value of that and the notion of that, that's going to be another, trend to some degree, or not necessarily trend, but there's going to be increased demand for it because there's great value in it.
So it's like when you're out there collecting to get the mycorrhizal spores, you're also getting lots of the diversity of the bacteria and the fungi and protozoa and the nematodes as well. So, don't forget that you want that diversity of other material. Just when you're going into a forest or any place, just take small amounts, just take pinches, just a little bit is all you need for the inoculum. It just terrifies me when I go into a forest system and, a quarter acre of the forest floor is just completely ripped up because somebody's been searching for truffles or something like that. And it's just, okay, we're trying to destroy all of our resources. Be respectful of the environment because the existing system has to survive. So we can come back and find all of that diversity.
And part of learning
it's-
how to forage mushrooms, there's also ethical guidelines that go along with that often are rooted in indigenous principles of how to live with the land.
It just occurred to me, I just throw it out there, just crossed my mind as you were saying that. So yes, of course, thank you. And yeah, same thing. You don't need to get crazy amounts of soil to get spores. You can get pretty high concentrations, or just start with whatever you get from a really small collection. And then in one cycle, you can really quickly amplify that. And then you start to have a stock of these indigenous arbuscular mycorrhizal strains. So yes, thank you. But the thought I was having was sort of in a similar way to that notion of, if you, hunt an animal, ideally, most respectfully, you try to use as many parts of that animal as you can in whatever way, the hide and the bones and whatnot. If you're collecting soil, if your primary intention was just to get the AM spores, maybe for me going forward, it's like, well, I'll also make a batch of compost tea with the rest of that soil that, isn't the spores or whatnot. You're separating them out, but there's still all this other value and abundance there. Yeah.
Yes.
And speaking of succession and complexity and diversity, we've been talking a lot about compost, but I think the term has so many different notions for so many different people. I'm curious to hear from a mycologist perspective, what do you hear when I say a really beautiful, mature compost? What does that look like to each of you?
Well, do you want me to go?
I would love to.
Well, I'm very much involved in so-called composting in Australia. All of the food waste is now going to major recyclers, and they're putting it out as compost. Well, as far as I'm concerned, it's reduced waste. It's been pasteurized. It's still hot. It's not compost until it's cooled down and it's gone through a maturity period. And that can take some months. What's really concerns me with the hot material that goes out is that there are volatiles coming off which are actually toxic to the people who are working with them, but also toxic to the soil. Now, what also disturbs me here in Australia is that these big processing companies put the material out very rapidly because they don't have storage space to actually go through maturity. And our Australian standard, which is called the 4454 standard that these companies work to, would you believe that they're making compost, they're calling it compost, and the 4454 standard does not mention biology. So how can this be compost? It's reduced waste. And so part of the work that I'm going to be doing with this new grant is to look at the maturation period to actually talk about real compost, not reduced waste, and to try and see if somehow or other we can get into the 4454 standard, which is the national standard, something that mentions biology. So how can you call something compost if it doesn't have any biology in it? It's reduced waste.
Yep,
and so this is my dilemma in Australia. I'd be interested to know what Elaine's dilemma is in the US with what they call compost. It's not compost, it's reduced waste. Call it what it is.
We have exactly the same problem. You go to a composting company, and we always tell people, if you drive up and you park your car and you just get flies all over the windshield, and you don't really want to even walk out, just put the key back into the ignition and drive off, because do not stop here. Now you go to a composting operation and okay, no flies, great. So you get out of the car and you just about pass out from the smell of ammonia. Okay, crawl back in the car, leave. It's got to smell like compost. It should smell like a good forest floor. You need to know that they got the temperatures up high enough, long enough, but then you turn in order that you get the stuff on the outside that didn't kill the pathogens. That's got to go in the middle. And what was in the middle, so you've got to turn it there. You cannot make good compost if you don't turn it. Now you only have to turn it twice. Well, you turn it once when you put the pile together, and then 2 more times. And that should take you through the whole portion of that pile should have composted. Yeah, you're basically pasteurizing it. Yeah, we like to make sure that it's been, it's food standards that we're really following. When we're talking about it has to be above 55 degrees Celsius or 131 degrees Fahrenheit for 3 full days, or it's got to be above 145 for 48 hours, or it's got to be above 160 for 24 hours. You're doing what you do in food microbiology to assure that there are no E. coli, no Salmonella, Shigella, Pasteurella, all the nasties that human beings are gonna get sick and have a very unhappy time if you consume them. So we want to make certain that we don't have any of those problems. And then we want to see that you have a minimum of 135 micrograms of bacterial biomass, 135 micrograms of fungal biomass. We want to see, 10,000 protozoa per gram, or, somewhere in that range. A little more wishy-washy on that one. And then you want to see at least 100 good guy nematodes. No root feeders, thank you. They should all be beneficials. And that's the minimum level. You go up to a composting operation and you bring your microscope along and you take a quick look. And if you see organisms moving and zooming all over, yes, this is a good compost as long as it's good guy organisms. And then if you look at at another sample and there's nothing moving, there's no fungal hyphae, no protozoa, no nematodes, well, that's reduced waste. So pretty easy for people to tell. And then when you look at the balance of the fungi to bacteria, you want to work on that a little bit to make sure that the fungal to bacterial biomass, the ratio between ammonium and nitrate, are going to be promoting your plant.
Yeah, and as that compost matures after it goes through those thermophilic cycles, I'm curious to understand a little bit more about what is going on inside of the fungal world, because I know that's the time when really our fungal numbers are booming, and also the diversity is increasing because the temperatures are cooling and that food resource has now become rich and available. Does anyone feel compelled to share any more about what's really happening on inside of the fungal mind at that point?
Well, actually, in some cases, I would suggest we don't really know completely. We know that the more diversity that we have there, we have the fungal population that has different enzymes, has different reasons for being in a particular situation. And so this is why you see the succession. You can see it in the higher mushrooms, and then you can see it in the micro fungi as well. The biochemistry of these organisms is often very different, but also within a single genus, you can have species that have different mechanisms for whatever it is their role in the environment at the time happens to be. And so I'm thinking of the work that my PhD student just completed did on trichoderma. He was looking at trying to find species of trichoderma that produced metabolites that would knock down a particular fungal species that was a problem with lettuce around the world, particularly in winter. It was a winter active organism. Now, traditionally, the fungal commercial products have always been warm temperature species and weren't active in the winter. And the commercial products that were necessary had a bacterial component added to them. But during the course of his PhD, and certainly with other trichoderma workers around the world, particularly in Spain and Italy, we're finding a lot of cold climate species. So in terms of how their biochemistry works, a lot of it depends on what they're doing at the time within a particular environment, within a particular pH situation, within a particular access to nutrients. So when I'm looking at it, I'm looking at a specific point in time, and therefore what that organism is doing at that point in time may well be different from what it's going to be doing at another point in time, given access to a different nutrient source.
Absolutely.
I
popped
think
up this related question too. I'll give you the mic back, Elaine, but could you please talk about the chemistry of fungal pigments? So yeah, there's so much going on biochemically in that time.
Well, a lot of the fungal pigments you see, particularly in the Dermatiaceous group, you'll find a lot of them are black and brown. You'll also find their spores, for example, in groups like Stemophylium, Alternaria. They have very dark, thick-walled, but you'll find a lot of those are also exposed to UV light. So where you've got organisms that have an exposure to sunlight, you'll find them often with a much darker pigments than those that are more protected, I guess, from sunlight. And I'm thinking my very favorite organism on this planet is Botrytis. And I have many isolates of Botrytis that go from absolutely haline, absolutely colorless, through to the most beautiful pinks. And so, but they all come from different environments. So the more heavily pigmented are those that tend to be more exposed to ultraviolet light.
Or other tough conditions, right? The
tough conditions generally, yes.
Yeah, or, a microarthropod trying to penetrate those walls. Wonderful.
Yes, protection.
Do you have any wanderings around fungal pigment chemistry or other fungal chemistry while we're on the topic?
Yeah, what Mary's just talking about, kind of where my mind goes, is not, I don't use the term pigment in that scenario, but more fungal melanins. So it's sort of like a big question about the importance of fungal melanins and their application. When I first looked in this years ago, I thought melanin was just like there's one type of melanin. It's a whole class of compounds, and fungi produce all kinds of melanins. And we find them as kind of a theme and, present in sort of the darker pigmented or maybe thicker walled, as well as the more physiological attribute, but pretty common in the more resilient fungi. Is it just as a means of protecting from the UV exposure? Is there some other benefit or some sort of side effect that comes with that? As far as I know, there's no conclusion, but there's definitely a correlation. And there's some papers, I think there's even a book I might have seen and want to get called Fungal Melanins, because it's just like, what's up with this? This seems to be a common thing. It's another huge area of fungal chemistry that we should probably look into. It's kind of as much as I can say. As far as
that's wonderful. Yeah, those fungal melanins I've also read are a large component of, because they are so stable and resilient to those abiotic conditions, a large proportion of the molecules found in humus. And humics are from degraded cell walls because of those melanin components that don't break down easily. And so that is that long-term stable carbon in the soil as well, is coming from those melanins.
So the fungal melanins color the soil. I hadn't
heard
that
crazy?
Connection. That's rad. As far as mushroom and their spore colors, just to take it a step further with the pigments, there's no consensus on why even a mushroom, is as complex as it becomes, just to spread its spores, let alone why are spores pigmented. There's different hypotheses, but there's no consensus.
There's not enough data on it, really.
Yeah.
And I think like when we're looking at different enzymes and we're looking at different abilities that can be expressed, we'd have to be looking in the DNA and start to understand what each color or pigment or enzyme was coded for in the DNA of that organism to really understand their breadth of ability.
Mm-hmm.
And until we start to connect color with the DNA, messenger RNA, etc., I don't think there's really much chance of figuring out the connection. What's the function of the microorganisms and that for me is a more important question than taxonomically identifying where something goes in the taxonomic structure.
Well, that sounds like a great question for you and Mary to talk about over another glass of Shiraz someday, it sounds like.
I think it'll happen in the not too distant future. I'd love to be helping you, Mary, with your new grant. Just let me know how I can help.
Absolutely, I would love to talk with you about it. It's an opportunity to actually get some quantification, and you and I know that it's possible. But outside that, I keep hearing from researchers who don't include me in their work that there's no way of quantifying the microbial world. Well, you and I know it's there. We're doing it. We've been doing it for years. So this is an interesting opportunity for us over the next 2 or 3 years to maybe collaborate on that quantification and actually try and get somebody in Australian people who make up the standards that compost actually requires microbiology. Oh wow! Surprise, surprise.
Yep. Pretty easy set of trials where you have dirt plus a small amount of the good compost and show the growth over time, and then start with another one where you have just compost and show the growth rates along with each of the stages. So
it's fundamental science, Elaine.
Yes, it's seems unimaginable that people haven't figured out that they should be doing these kinds of experiments already.
Yes,
but that's okay.
Yes.
That means all of us can become very elite because we thought of it first.
Like decades ago.
Your collaborative relationship is an inspiration as a model for how to engage in science in the world and community, and it actually exemplifies what the final question that I had in mind for each of you as we wrap up. And that is that I often say that if we want to work with fungi, we need to act like fungi and behave and think like fungi. So I'm curious, what does that mean to each of you?
Peter?
I love the fungal human, relationship metaphor mirroring potential. Fungi being so successful and have been around for so long, and from an evolutionary standpoint, we essentially descend from them. I think there's definitely something to learn from their habits, even though they are a totally different branch on the tree of life. There's the obviousness of, say, the mycorrhizal, interconnectedness notion and the webbing. And that's where sort of I came into this concept many years ago of respecting fungi, learning from them. We should all be more distributed, share information, share resources. That's mirrored in resiliency models of how cultures and anything bounces back is through hyperconnectivity. So there's that as, just a netted thing in nature that's doing that really well. I've, in more recent years, taken that a step further and recognizing that netted culture is comprised of all the individual hyphae, and all the tips of those hyphae is essentially an individual that's doing its own thing to navigate its tiny little piece of the world and learn from it, dynamically engaged, release the knowledge, the enzymes that it has that to navigate and survive in that moment, learning from the environment, and then feeding that information back to the larger culture. This is how mycelium functions. And so, we're connected. We're sort of individuals, of course, in our day-to-day, but we are inevitably tied to not only the immediate culture, but all of our ancestry and the web of knowledge of time. And we send that forward, and we're spreading our own spores of knowledge in that way. And we are affecting and in relationship to our ecology, and not separated from it, of course, just like fungi are, and sculpting that. So, and just seeing how they're doing it with not only, looking out for their own needs, but sort of they've evolved, we can say, to, everything is working in concert. And so their effects, though we often talk about individuals in an ecosystem as sort of looking out for themselves, obviously everything works together. So we're often looking out for ourselves and maybe forget about the others. So having those notions, and not just our own lifespans, 7 generations lifespans, geological time spans, and how those effects ripple out.
Thank you.
Well, I liked what I'd read just recently. Sort of between 600 and 500 million years ago, the first plants that grew on Earth were actually ferns, and they had really next to no root system. And there's now a theory that maybe the mycorrhizal fungi and the fungi that were in the soil actually provided the first roots for the plants that came above the soil surface, and provided them with nutrients, and therefore started oxygen cycling and whatever. And so I just think with absolute humility that here's this wonderful kingdom that's been here for so long, doing so much good. And in the last couple of 100 years, we've gone all out to try and destroy it. Maybe not knowingly, but certainly that's what's been happening. And that if we want to continue to feed 9 billion people and rising on this planet, then if we start to consider, as you've just been saying, Peter, that the fungi are such a diverse kingdom, but also such a sharing kingdom, that if we looked at the way that sharing and that regulation and that looking after self and looking after others actually occurs, then humanity maybe has a future on this planet. But I certainly don't feel that if we keep going headlong into destroying the soil, saying that we have to clear forests to grow more fruits and vegetables and whatever to feed the population, we don't need any more soil. We don't need any more food. We've got plenty of food. It's not being distributed correctly. And also, if we stayed with the soil that we have and actually used it correctly, we don't need more soil. We can feed the planet, but we have to do it with a little bit of care for the soil. And I say to my clients, my farmers, my friends, farm your most important resource, and that's what's below the soil. That's what you should be farming, because anything you grow above the soil will grow brilliantly if you farm your soil. So look at soil as your greatest resource, not what you can produce from it.
Yep. And we need to have growers realize that as you go through succession, you just have to get the right balance of the organisms so that you're selecting for the plant to be as healthy as possible. If we follow Mother Nature, who's been doing this kind of production for the last, 3.5 billion years, or if we're going to talk plants, for the last billion years, that's plenty of time for her to have figured out what these relationships need to be. We need to stop being so arrogant that we know better than nature. That's, no wonder I sometimes sit back and wonder what nature thinks about us. And just, I needed to get all of that carbon that was down there in those wells, in those petroleum fields. And I needed to get all that carbon back up here on the planet. And I didn't know what organism would be able to do this for me, but no, I found them, people. Now that all of that carbon is pumped up here, what does she need us for?
Yes. Yeah.
So we better pay attention or we won't be here.
Exactly.
So on that really happy note.
Yes.
Conclusion, the end. No, it really is such a hopeful thing, especially to hear each of you and your different perspectives, and to hear the individual work that you are. You're each your mycelium tip out there that's sensing and getting grants and bringing them back in so that we can streamline and bring the rest of our human community and other non-human communities along with us. And yes, the fungi are a masterclass in humility that I'm continuing to learn from. So the grant thing is what made me go to private business, because I am so happy for you for having that grant, and that's such hard work. That is so difficult to get the money. So please involve us. We would love to.
Absolutely.
Work with you.
It's a given.
Peter, any way that we can support you, let us know. Elaine, any way I can support you? Always, I'm here. I think that's the way to be, right?
Yep, we need to get Peter on board as well. So all hands helping.
Awesome. Well,
all
thank you
all the
so
people
much.
out there in the audience too.
Indeed. Thank you all for being here and for thinking about fungi and knowing that this is a really stimulating area and discussion, and keep on cultivating and sporulating, right?
Well, I'm happy. I'm happy for people to email me. It's better than calling me. Look at my website, which is agpath.com.au. A lot of information is there, but always feel free to contact me on an email.
Okay, shall do.
Wonderful. Okay.
Thank you.
Thanks, everyone. Thank you so much.
Thank you.
It's a pleasure to see you.
Bye-bye.
Ciao.
Yeah, thanks, everyone.
Bye.
Don't forget to click that like button, subscribe to our channel, and ring the notification bell to stay updated with all our new videos.